we have discussed all these things, and we were discussing regarding the Sudan black staining, right? Yeah, perfect. So is it visible to all of you?
Yeah.
Okay, I'm going to start. So I was discussing regarding the techniques which we can use for the detection of the lipids within the microalgae and the cyanobacteria, and for that we can use the Sudan black stain. And as the name represents that the black color would be appear for the detection of the lipids within the microalgal cells. And here are the results which have been shown for the unicellular and the filamentous microalgal cells. Now come towards the another technique which is known as confocal laser scanning microscope, and it was used for the detection and the localization of the intracellular lipids. And for that detection, I used the Nile red and the body P dyes, and here are the conditions which I used because these are the dyes and they have to show the some fluorescence when they combine with the lipids. So they will show fluorescence at the specific excitation and the emission wavelengths, and these wavelengths vary for the both of the dyes. And if we have the more number of the droplets, then the more fluorescence was detected. And the volume of the lipid droplets, they was also measured via using the software known as the image. So here are the results of the filamentous and the unicellular microalgal and the cyanobacterial strains. Here you can easily see that among these trichomes and the filaments, the yellow lipid color, the yellow color is showing the lipids which can be fluoresced by the Nile red dye. However, the green color is showing the fluorescence of the lipids via the body P dye. So both dyes show the different fluorescent colors for the lipids. Come towards the volume of the lipids which was detected from the software which I used for the detection, and that was the image J, and highest volume was obtained in form of the Lingbia, which is the filamentous cyanobacterial strain, and among the all unicellular cyanobacteria strains, microcystis showed the highest volume of the lipid droplets. And in the unicellular microalgal strains, glycosystis showed the highest results. Now come towards the another technique known as the Fourier transform infrared spectroscopy for the detection of the molecules, especially the functional groups present in the biodiesel. And as all you know that the ester group is present in the biodiesel, so that was detected via this technique. And here are the graphs showing the results for the ester group. Here are the some results which have been shown in the form of the graphs and the tables. Now come towards the another type of the biofuel known as the biohydrogen, and this hydrogen can be also used as the source of the biofuel. So for the production of the hydrogen, hydrogenase enzyme is present in the cyanobacteria and the microalgae, and which they can be used as a source of the energy. And I detected this hydrogenase enzyme and the production of the hydrogen among the 8 different conditions. So here you can see that the differentiating points were the Bg11 media having the nitrate, and Bg11 0 means the absence of the nitrate in the media. The second difference point was the presence or absence of the glucose, and third difference was whether the strains were present in the light or in the dark. Here is some pictorial representation and glimpse of my results and my research work which I performed in the Uppsala University, Sweden. And here are the results and the peaks which can be shown for the detection of the hydrogen. And in the tabular form, I have also summarized my results. So here you can see that the 4 columns especially showing, the first column is showing that the strain is present in the dark and in the presence of Bg11 nitrate media. And the second column is showing the results for the dark and in the absence of sodium nitrate. The third column is showing the results for the dark and in the presence of the glucose and nitrate. However, the fourth column is showing the results when the strains were grown in the dark in the presence of the glucose and in the absence of nitrate. So these conditions were used for the detection of the hydrogen among the different cyanobacterial strains. And the same results same procedure was used for the darkness and the in the presence of the light. So here you can see the results of the unicellular cyanobacterial strains, and the highlighted regions are showing the highest results which was obtained in the different specific conditions. Now I think you can better see and read that what does these columns means. Here are the results for the unicellular cyanobacteria in the presence of the light, and unicellular microalgae in the presence of dark, and unicellular microalgae in the presence of the light. So in this way, I use the 8 different conditions for the detection of the hydrogen in the various environmental conditions for the production of the hydrogen. So the summary of the results regarding the biohydrogen is that among the dark condition, the highest results was obtained by the leptolimbia for the hydrogen production, and in the light, oscillatoria shows the highest result. And among the unicellular strains, Cyanococcus species showed the better results in the dark as well as in the light. However, in the unicellular microalgal strains, Chlorella showed the highest results in the dark. However, Gliocystis showed the high result in the light. Now come towards the mutational analysis, which I performed via the UV mutational analysis. So I gave the UV for the different time periods, which was the 5, 10, 15 minutes. However, I also experiment for the greater than 15 minutes, and that was the lethal condition for the growth of the cyanobacteria and microalgae. So I decided to just use the 3 different conditions for the detection of the for the effect of the UV on the production of the hydrogen production from these microorganisms. So these highlighted regions are showing that these strains showed the highest hydrogen production even after exposure with the UV light. So here are the results for the unicellular cyanobacteria, unicellular microalgae, and the summary of the biohydrogen production, even in the presence of the UV mutational analysis. So highest result was obtained in case of the leptolimbia, and among the cyanobacteria, Cyanococcus, and in the unicellular microalgae, Chlorella showed the highest result for the production of the biohydrogen. So this was the end of the second part of my research work. Now come towards the third and last part of my research work regarding with the production of the third type of the biofuel known as the bioethanol. And the same conditions were used which I used for the hydrogen, that in the presence of Bg11, in the absence of nitrate, with the presence or absence of the glucose, and as well as in the light and dark conditions. And same you can see the different graphs which was obtained for the detection of the ethanol in the from the cyanobacteria. And here the summary of the results have been summarized in the form of tables for the different conditions, same which I discussed for the biohydrogen. The same pattern was followed for the detection of the ethanol, and the conditions are same. Okay. And so the highlighted regions are showing the maximizing results in the specific conditions. Now come towards the summary, that in the filamentous, among the filamentous cyanobacterial strains, leptolimbia showed the highest bioethanol production, which is 403 mg per liter in the dark. However, 291 mg per liter was obtained in the presence of the light. So if we will see the all results on this page, so maximum result was obtained in the form of the unicellular cyanobacteria, Cyanococcus, which showed the highest result in the presence of the light. As you can see that 2221 mg per liter of the bioethanol was obtained from this mini creature. So you can see that how much interesting these microorganisms are. So same mutational analysis was performed for the detection of bioethanol from them, and the highlighted regions are showing the maximum results. And you can see this is for the unicellular microalgae, and here is the summary of all the results. And after giving the UV mutations, so highest result was obtained in the case of the unicellular microalgae Chlorella. Now come towards the final conclusion. Cyanobacteria and microalgae are the source of the multiple biofuel productions, which I have already discussed with reference to the biodiesel, biohydrogen, and bioethanol. And the maximum biodiesel productivity was obtained from the leptolimbia, which was obtained from the northern area of the Pakistan. However, for the detection of the lipids, polyunsaturated fatty acids were obtained from the unicellular cyanobacteria. And the highest volume of the lipid droplets were obtained from the Limbia, which was obtained from the northern area. Now come towards the second type of the biofuel. So for the biohydrogen production, microalgae chlorella showed the highest amount of the hydrogen. However, the bioethanol was obtained in the maximum amount from the Cyanocystis, which is a type of cyanobacteria. So if we will summarize these results, so filamentous cyanobacteria are good biodiesel and biohydrogen producers. So they have the ability to produce these two types of biofuels in a better amount. However, unicellular cyanobacteria is a good source of the bioethanol production. So here is the summary of the results again for the multiple biofuel production strains. Like upper four strains are showing they were obtained from the northern areas. However, the below four strains they were obtained from the Punjab area, and these in total among 65 strains, all 8 strains showed the highest amount of biofuel, either in the form of biodiesel, either in the form of biohydrogen, or either in the form of bioethanol. So future plan is that to make the advanced molecular techniques to get inside the mechanisms and pathways which we can use for the better production of the biofuels, how we can modify it, and as well as what approaches can be used for the increased production of the multiple biofuels. And by developing the certain conditions, how we could be able to produce the multiple biofuel by single strain, that from by utilizing just a single strain, how we can obtain the all three types of biofuels in the higher amount. So this page or these future plans we are working, and the fourth generation is basically regarding with this slide, that how we can modify the microorganisms, how we can modify the pathways, how we can modify the biofuels, and how we can modify the single strain for the production of multiple biofuels. So take home message is go green, and by utilizing the biofuels, we would be able to use these biofuels for the vehicles, and we would be happy in the end. So here is the just glimpse of my article, which I have already published, and you can see and you can search anywhere. So many thanks, and the floor is open for the questions. Yes, please. Does anyone have question, or anyone who wanna ask anything? You are free to ask any type of question. I am available, and I would be happy to answer you. Okay, there is a question in the chat. Okay, there is a question that after how much these microorganisms will produce the maximum lipids and biofuels. So in comparison to the first and second generation of the biofuels, they produce these type of biofuels too much earlier than the first and second generation. Like while utilizing the plants, in comparison to the plants, we have a very short time period for the production of biofuels. So there is some difference regarding the whether we are obtaining the biofuel in the lab, or whether we are obtaining the biofuel at the higher level, as I have already performed my research work in the natural environment as well. So in the nature, they are more prone and they are more happy to produce the biofuels, and in actual within the 2 to 3 months, we can get the biofuels from them. I hope you get your answer, Abdul Majid. Anyone else? Anyone else? Okay, there is another question. Is the biofuel environment friendly? Yes, when we obtain this biofuel, so it doesn't produce any type of nitrates or nitrites in the environment, and it is too much environment friendly. So we can use this as a source of the biofuels. However, now there is a focus towards the modification of these microorganisms for the better production of the biofuels. That's why the genetic engineering technique is in use for these microorganisms for the production of biofuels in the higher amount. No, Abdurrahman, this is user friendly, so there is not any possibility of the global warming by using these microorganisms. As you can see that already these microorganisms are present in the nature, and we are not specifically growing, and they are not producing or having a great effect regarding the global warming. As you can see that where there is the moisture and there is the water, so these microorganisms naturally grow in the environment, so they are not contributing an effect regarding the global warming.
Dr. Sara, we do have one more question there.
Okay, let me see. Okay, wait.
Okay, do this biofuel has been produced through microbes by any countries until now?
Sorry, do these microorganisms?
Do these biofuels have been produced through microbes by any countries until now? That is the question.
No, let me see because there's some voice distortion. Is it mentioned in the chat box?
Yeah, I get a,
Let me see just once.
that
Okay,
is
okay. Is the Bible having produced through microbes by any countries until now? Yes, yes. Especially these biofuels are producing in the higher amount in the Brazil, and this technique can be used anywhere in any country because simply you just need these microorganisms, and just you need to know the tactics that how to deal with these microorganisms, because these microorganisms are not similar in comparison to the bacteria that we can easily handle those, because as we have the ability that we can use different bacteria in other research purposes. However, these microorganisms are very, you can say that they are moody, and if you know the tactics of these microorganisms for their growth, then you can produce the highest amount of the biofuels from them. Yes. As I am adding some more thing that as I went to the Sweden, so there they are using these microorganisms for the especially for the production of biohydrogen and for the production of the bioethanol. But my focus was towards the biodiesel as well. So I did my research work over there regarding the biodiesel as well. So in the many countries, this task is already under consideration and they are performing that, but the special need is that how we can utilize these for the better production of the biofuels. And here's another question: why mutated organisms produce more biofuel? Yes, and you know that these microorganisms have the ability by the nature that they are present in UV light, right? Anywhere where the sunlight is present, anywhere where the moisture is present, where there is the water, then these microorganisms have the ability to grow. So that is why they have been mutated naturally due to the continuous facing of the UV light. Okay, so that's why these microorganisms produce the better biofuel in the presence of the UV. Did you get your answer? Yes, it might be because I didn't focus or I didn't have much time for that to observe the mutational analysis at the genomic level. So in the future, I have a plan that I will move towards this aspect as well. Right now, I'm not sure, but maybe.
So this is it for today, maybe. So hope everybody has got a lot of information about microalgae and biofuels as well. So it would be a great alternative source of energy in this energy crisis conditions, and hope we can do much research and development work in this as well. And I want to thank Dr. Sara Junaid for presenting it wonderfully, and hope we will be doing much more work in the future as well. So, kind of collaboration needs to be done in the future as well. So, thank you for all the attendees as well, and then the person who have raised the question as well. So, hope to see you all in the upcoming sessions, upcoming webinars and workshop as well. We are soon organizing one workshop regarding in silico primer designing. So, hope to see you all there as well. And this is it for today. Thank you.